Hello, and welcome to this presentation. My name is Vijay Ramchandani, and this presentation is an overview on alcohol metabolism. I would like to acknowledge my colleague, Dr. Soundarya, for her help in developing these slides and materials. First things first, why should we even know about alcohol metabolism? Why is it important? As this picture depicts, there is a huge variability in the vulnerability to alcohol use disorder across the world. This makes us wonder, why are some people predisposed to alcohol use disorder? And how might the pharmacology of alcohol be related to this? To understand this, we need to understand the variability in the pharmacodynamics, or simply put, what alcohol does to the body, which depends on its systemic concentrations over time, or its pharmacokinetics, which is what the body does to alcohol. That is to say, after consumption, what are the alcohol concentrations that result in the body and in the brain, and how that is determined by alcohol's absorption, distribution, and metabolism. This is hugely variable. Let us see how. Here is some data from a study where 12 healthy volunteers were provided a conventional, single, standard oral dose of alcohol, and breath alcohol levels were recorded as a function of time. The x-axis represents the time duration after alcohol, and the y-axis represents the blood alcohol levels. And as you can see, there is huge variability in the blood alcohol time curves for these individuals. And when you look at the entire profile, you see this tremendous variability in exposure. Peak alcohol concentration range is extremely wide, ranging from 20 to 70 milligrams per deciliter. And you can also see the variability in the overall profile. Given this range, it is critical to understand the underlying sources of variability in how alcohol is processed by the body, which in turn will inform us about variation in alcohol response or pharmacological effects, and how that drives vulnerability in risk of alcohol use disorder. This slide shows an overview of the processes involved in alcohol pharmacokinetics. Here we are depicting the absorption of alcohol through the gastrointestinal tract into the liver for first pass metabolism, and then distribution into body water. From there, alcohol is metabolized back in the liver, as well as in some other organs, but also excreted unchanged in the kidneys, lungs, and sweat. And in this presentation, we are going to review the processes of consumption, absorption, distribution, hepatic oxidation, and elimination, and their associated variability. And then at the end, we will talk briefly about the role of metabolism in hangover, as well as, importantly, sex differences in alcohol pharmacokinetics. Let us begin with how various factors associated with alcohol consumption affects its variability. What we drink matters. This figure depicts different blood alcohol curves derived from the consumption of different alcohol-type beverages. Spirits, wine, and beer, depicted in different colored curves. The X is the time point since alcohol consumption, and the Y-axis represents the mean ethanol concentration with standard deviations. And as you can see, that absorption is slightly faster and higher following the consumption of spirits relative to wine and beer. All of these administrations were done in the fasted state. This figure depicts the data after rapid consumption of 4 doses of alcohol in the fasting state. This is from another study in healthy men. And as you can see, the equivalent of 1 drink, 2 drinks, 3 drinks, and 4 drinks brings alcohol levels up close to above the legal limit after the consumption of 4 drinks. And you can see that the escalations in concentrations follow a almost linear pattern, followed by a peak that occurs somewhere between 30 minutes to an hour after consumption, and then a zero order or pseudo-linear decline. The breath alcohol increases, as I mentioned, seems to be proportional to the number of drinks. And as you can see, this is under the fasted condition, so the absorption is relatively fast. So the dose, of course, is a major determinant of absorption and therefore pharmacokinetics of alcohol. So absorption is a first order process for alcohol, and so the concentration gradient that results from a higher dose rapidly consumed will also result in rapid absorption, resulting in higher peak concentrations. So once it's in the gut, how is alcohol absorbed? Where is it absorbed? And what results in that rapid absorption? And how does it contribute to variability? So alcohol is readily absorbed from the GI tract, as I mentioned, after consumption. This starts in the oral cavity, but a very small proportion there, but progresses as we descend down the GI tract. As you can see here, the large majority of the alcohol is absorbed from the small intestine, much more rapidly than the stomach. The small intestine is rich in structures called villi, and these are the projections on the inner surface of the small intestine, increasing the surface area for absorption. Once alcohol is absorbed, it spreads freely into all the body water spaces. Alcohol does not bind to fat or protein, so there's no plasma protein binding reported for alcohol. As I mentioned, the rate of absorption of alcohol is highly variable and depends on a number of factors, some of which we have already looked at: the dose, the alcohol concentration of the beverage. Food intake can also have a major effect. It can affect gastric emptying, gastric metabolism, which in itself is a source of variability. And then hepatic first pass metabolism can also contribute to this. In this study, alcohol administration was conducted using intravenous and oral alcohol administration, and the oral alcohol was given in the fasted and fed states. And these are the resulting alcohol concentration time profiles. So again, you're looking at the blood alcohol on the y-axis and time on the x-axis. What you can see is that the highest blood alcohol was achieved after IV alcohol, indicated by the red arrow and text, whereas the oral alcohol under fasted condition, indicated by the blue arrow, was significantly lower. What you can also see is that after meal consumption, whether being fat, carbohydrate, or protein, resulted in an attenuation of that alcohol level as a function of time. And so under fed conditions, there is a lower availability or absorption of alcohol. What we don't know when alcohol is given orally with food, whether there is a slowing or delay in absorption, or there is a alteration in elimination that might contribute to the lower levels seen after food intake. If you compare the peak levels that occur after oral and IV, you can appreciate the difference in absorption and first pass metabolism, which is the metabolism of alcohol as it goes through the first pass of the liver before it enters the systemic circulation. And so with IV alcohol, of course, the alcohol is going directly into the system, so you get a faster, higher peak. But after fasted state, in oral administration, you do get an attenuation of that. You can also see that metabolism is also slower compared to the fed state. The second image here on the right is from a study where IV alcohol was administered to individuals under fed and fasted conditions, and this was done to isolate the effect of food on elimination versus absorption. So what you can see here is the lighter gray bar is the alcohol elimination rate under fasted, and the darker gray bars are the alcohol elimination after high fat, carb, and protein meals. And again, here you can see that there is about 25% increase in elimination rate. And so with food, what is happening is there's a slowing of absorption, but there's also a speeding up of metabolism. And together, they seem to contribute to the lower availability or exposure to alcohol after food compared to the fasted state. Another factor that affects alcohol absorption is gastric emptying, and a good model or approach to study that is using bypass participants. And so this figure compares the breath alcohol level as a function of time in patients following gastric bypass who were given alcohol administration versus controls. And again, what you can see here is when there is gastric bypass surgery, there is faster gastric emptying, and essentially the alcohol goes directly into the small intestine, and you can again see a higher exposure or availability of alcohol because there is no gastric metabolism or delay due to gastric emptying. Interestingly, it is noted that in these populations that undergo gastric bypass surgery or bariatric surgery, there is an increased alcohol consumption and a risk for alcohol use disorder. And it is suggested that these higher alcohol levels might have more of an effect on risk for alcohol use disorder. It's also suggested that this surgical procedure can alter ghrelin signaling, and ghrelin being a GI feeding neuropeptide, which can stimulate alcohol intake and then can lead to higher consumption in these patients. So let us move now on to distribution. As we know, alcohol is a small polar molecule and rapidly and readily distributes in aqueous spaces. Thus, distribution to organs and tissues depends on the water content. It also depends on the blood flow and tissue mass. So greater the blood flow, higher the distribution. Greater the tissue mass, greater the distribution. Alcohol readily passes through biological membranes, however, it is insoluble in fat. The volume of distribution for alcohol is equivalent to total body water, which constitutes approximately 55 to 60% of total body weight. Sex differences in total body water is one of the main factors underlying differences in alcohol pharmacokinetics between males and females, which we will discuss in more detail a bit later. Moving on to alcohol metabolism. As I mentioned, alcohol is metabolized primarily in the liver, although there is metabolism that occurs in other tissues. There are two pathways for metabolism. The first, most predominant one is the oxidative pathway shown on the left. The first main enzyme that metabolizes alcohol is ADH, or alcohol dehydrogenase. This is typically seen in the cytosol of the hepatic cells, and then by aldehyde dehydrogenase, or ALDH, which usually occurs in the mitochondria. There are other enzymes involved in these oxidative pathways, possibly involving the cytochrome P450-2E1, and also an enzyme called catalase, which may be present in brain tissue as well. There is also a non-oxidative pathway where alcohol combines with fatty acids to generate fatty acid ethyl esters, phosphatidylethanol. These are important potential biomarkers for alcohol use because they are excreted in the urine, they can be measured in urine and blood, and they last for several hours to days after alcohol consumption, and so they may be clinically relevant biomarkers. So as we can see in this figure, the ADH pathway is primarily in the liver, and the non-ADH pathways appear to be in the brain. Importantly, generates acetaldehyde, and acetaldehyde is an intermediate of alcohol metabolism, which does not accumulate in the liver or in the tissue and is rapidly converted to acetate. However, there's interesting recent data. There's been data for a while, but with interesting recent data that has kind of tried to highlight the role of ALDH2, or aldehyde dehydrogenase, and importantly, acetaldehyde as an intermediate marker of alcohol's effects. And so in vitro and in vivo studies by Jin et al., they examined distribution in the brain and found ALDH2, which is the aldehyde dehydrogenase enzyme expressed in both cerebellum and astrocytes in the brains of humans and mice, and that there was a mediation of this ALDH2 in the production of acetate, resulting in cellular and behavioral effects. And this kind of adds to the evidence that has been accumulating for a few years now that acetaldehyde in the brain is locally produced and may be contributing to some of the effects of alcohol. So in the liver, within the hepatocyte, as I mentioned, ethanol is converted to acetaldehyde by acetate, and that occurs in the cytosol, and then the acetaldehyde enters the mitochondria where it is converted to acetate via aldehyde dehydrogenase, or ALDH. And this metabolism is the primary pathway. The acetic acid or acetate that is produced is finally excreted either unchanged in the urine or converted to acetyl coenzyme A and then enters various energy cycles. Oxidative pathways can also generate reactive oxygen species. As we know, acetaldehyde being an aldehyde and a reactive molecule can also cause that. Non-oxidative pathways are, as I mentioned, fatty acid ethyl esters or phosphatidyl ethanol, and they may also play a role in some of the toxic effects of alcohol, although that has been less clearly understood. So more than 50% of the variation in alcohol use disorder is contributed by genetic factors. And this brings us to this major subtopic in alcohol metabolism. Genetic differences or genetic variations in the genes that code for these two alcohol metabolizing genes, the ADH and the ALDH, those genetic variations seem to produce alcohol metabolizing enzymes with varied kinetic properties. This leads to different rates of alcohol elimination and acetaldehyde generation, as well as acetaldehyde elimination, can then ultimately affect the risk for alcohol use disorder. Let's unpack that a bit. So with respect to ADH, the general notion is higher enzyme activity of ADH, higher is the acetaldehyde production, and thus exposure, which then increases the risk for alcohol-induced liver disease. And major large genetic studies and meta-analysis have confirmed this. With respect to ALDH, lesser activity is associated with greater acetaldehyde, since less enzyme is available for metabolizing this acetaldehyde. And acetaldehyde accumulation can have toxic effects, leading to aversive experiences, and this may eventually end up with a protective effect against heavy drinking. This slide shows the different ADH genes and their associated enzymes and expression profiles. As I mentioned, there are multiple genetic variations in the ADH and ALDH genes, and the ADH variations result in multiple isoforms. So as you can see here, you have the ADH1A, the ADH1B, and 1C, which are the polymeric forms. These are all found in the liver. ADH4, which is found in the liver, but also in the stomach. ADH5 and ADH6 and 7 are less well described. They also have different metabolic capacities for alcohol or rate constants, and there's a wide variation in those. Within the ADH1B, there are two clinically significant or relevant variants. As you can see here, the ADH1B star 1 has a arginine in positions 48 and 370. The ADH1B star 2 is the variation where the arginine is replaced by histidine in position 48. And the star 3 polymorphism is where the arginine in 370 position is switched with a cytosine. And so with these variations, you get two different variants of this gene, the so-called beta 2 and the beta 3 variant. And this then leads to different isoforms of these enzymes that have different catalytic efficiencies for alcohol and therefore result in different levels of alcohol and acetaldehyde following exposure. So the ADH1B star 2 is associated with faster metabolism, which can result in accumulation of acetaldehyde, which can be toxic and unpleasant and result in a characteristic flushing response to alcohol. And this is manifested by a blushing or flushing appearance, a reddening of the face, tachycardia, as well as nausea. This can be highly limiting in terms of alcohol consumption, and so individuals with the star 2 variation tend to drink less. These are population specific, such that you see the star 2 most commonly in East Asian populations, up to 75% in individuals of Japanese and Chinese origin, and also slightly more frequent in Middle Eastern populations at 20%, but less than 4% in European individuals, but when present, it can actually have a particular. The star 3 variant is most common in African origin samples, and again, there's some indication that they may be protective against alcohol use disorder, but those data are less consistent. So this is a study where individuals with the ADH1B star 2 variation were recruited and given alcohol intravenously to measure alcohol metabolic rates. And what you're seeing here is the alcohol elimination rate for the three groups, the star 1 or the common variant, and the star2 heterozygotes in the reddish and the star2 homozygotes in the pink. And you can see here a significantly higher alcohol metabolism, about 20% in these individuals who carry the star2 variation. A study looking at the star3 variation in an African American sample was unable to show any significant effects, although there were previous studies to that effect have been reported, and this remains an area that is in need of additional research. With the ADH1C, there is thought to be an increased risk for alcohol-related cancer, although the metabolic effect or the pharmacokinetic effect of ADH1C seems to be less clear. And here you have this variation in these two positions, 272 and 350, as is depicted here. There are other ADH enzymes, and there are genetic variations such as those in ADH4, which have also been associated with alcohol use disorder and may also be contributing to its effects. The more clinically significant variation is in the ALDH2. And ALDH2, as I mentioned, or aldehyde dehydrogenase, breaks down acetaldehyde to acetate. And this is not localized in a single chromosome, and there are multiple genes that encode the ALDH superfamily. The one that is most relevant to alcohol is the ALDH2, and this variation in the 504 position, which results in a form of acetaldehyde that is mostly deficient at metabolizing acetaldehyde. And so this is prevalent at the rate of 12 to 41% throughout East Asia. Individuals can be heterozygous, which has a strong protection against alcohol dependence or alcohol use disorder. And homozygotes who carry 2 copies of this variant are almost completely protected from alcohol. And so, as you can see here, this was a study done in a Japanese sample where they recruited individuals with the different forms of the ALDH2 and gave them an oral dose of alcohol. And you can see here that there is a deep reduction in the beta 60 or disappearance rate for alcohol. It slows down in the ALDH2 homozygotes. But what you see importantly is this escalation in acetaldehyde levels 5-fold higher in the ALDH2 heterozygotes, and in the ALDH2 star2 homozygotes almost in 20-fold higher acetaldehyde levels. And so you can imagine these are going to be extremely aversive and make alcohol consumption nearly impossible in this group. And so the substantial impact of this variation on alcohol and acetaldehyde pharmacokinetics and the consequence of that on the risk for alcohol use disorder is quite apparent with this gene. The other enzyme, as I mentioned earlier, is the CYP2E1, and this is a cytochrome P450 enzyme that is also present in the liver. It seems to be inducible enzyme that kickstarts when alcohol is consumed in larger amounts or following chronic exposure. It can account for up to 10% of ethanol oxidation and a primary component of the so-called MEOS or microsomal enzyme oxidation system. The important attribute here is the inducibility, which can be up to 10-fold higher and could result in some metabolic tolerance to alcohol as well, along with reduced ALDH activity, and add to the risk of alcohol use disorder and, importantly, alcohol liver disease. As you can see also here in this graphic, CYP2E1 results in the formation of rapid oxidation species, which can then add to DNA adducts and can have adverse consequences there as well. So it's important to consider this enzyme, particularly once it is induced, in terms of its impact on pharmacokinetics, but also alcohol use disorder and alcohol liver disease risk. This is a summary of all the genetic variants involved in alcohol use disorder. And the top of the list, as you can see, and highlighted with a red box around it, are the 2 alcohol metabolizing genes that have been the most consistent and the most prevalent in terms of its risk, in protection of risk against alcohol use disorder. And in these populations, you can see that there is a reduction in vulnerability as well as craving. Another important factor to consider here is that the aldehyde dehydrogenase effect can be mimicked by a drug called disulfiram, and it's a drug that blocks ALDH2 and pharmacologically results in an elevation in acetaldehyde and makes the drinking extremely aversive, and has actually been used as a treatment for alcohol use disorder. And so this is another example where pharmacokinetic manipulation can have an effect on pharmacological response and risk. Finally, moving to the elimination processes. So these are processes of once alcohol is metabolized or alcohol that is not metabolized, where does it go? And so again, just bringing back this curve of the blood alcohol concentrations as a function of time following different doses, you can see that the decline of alcohol levels after the peak follows this pseudo-linear or apparent zero-order kinetic profile, while the actual metabolism, since it is capacity limited, follows something that we call Michaelis-Menten Kinetics. And while metabolism is by far the major route, about 2 to 10% of a consumed dose of alcohol is excreted unchanged and can be detectable in blood for several hours after consumption, as you can see on the left. And so a question that comes up is what represents the current brain alcohol concentration or the alcohol concentration at the site of action better, whether it is blood or breath? And really the answer is that breath alcohol concentration, because it more accurately reflects arterial concentration and what actually is what the brain is going to be seeing may be closer, particularly during the absorption phase and until it gets to peak levels. On the right is shown the three main routes where alcohol can be excreted unchanged. So through the kidneys up to 2 to 4%, through the lungs also another 2 to 4%, and in fact, and through the skin 1 to 2%, through the sweat and through diffusion. And the measurement of unchanged alcohol in the skin and the lungs has actually been used as a direct measure of alcohol exposure. So using devices such as the breathalyzer that measures unchanged alcohol excreted through breath, and recent innovations in wrist-worn and ankle-worn sensors that provide direct measures of alcohol exposure, which have leveraged these small but significant routes of excretion. Now, at the end of this presentation, I wanted to cover a couple of topics just related to the impact of alcohol metabolism. And the first is on hangover pathology. So recent studies have shown that alcohol pharmacokinetics may be associated with acute after effects such as hangover. So while hangover is not well understood, as we know, it is a range of physiological and psychological symptoms or effects that are seen following consumption of alcohol and after levels have kind of declined. But there may be a role for pharmacokinetics, even though there's not actually alcohol in the system. There may be effects of the alcohol metabolism that preceded the development of hangover. And so this recent study actually evaluated whether alcohol elimination rate or ethanol elimination rate, shown here on the x-axis in these panels for these individual subjects, was related to hangover symptoms. And what they found was, even though it's a small sample, a very consistent inverse relationship between alcohol elimination rate and hangover severity. And so this suggests that faster elimination of alcohol actually may be associated with less severe hangover symptoms, which may mean that slower alcohol elimination rates means there's more alcohol hanging around in the system and more generation of metabolites and other products that may be contributing to the hangover symptoms after alcohol consumption. Finally, in the last few minutes, I wanted to do a quick overview of the important, clinically significant and really relevant variable in alcohol pharmacokinetics, and that is, of course, sex differences. So there's been a really interesting recent meta-analysis that focused on alcohol and liver disease and clearly demonstrated that consumption of even one drink per day compared to long-term abstainers showed an increased risk for liver cirrhosis in women, but not in men. And similar effects can also be seen for other toxic effects of alcohol on tissues and organs, including the pancreas, the brain, the heart. And in all of these, greater pathology is seen following lower exposures in women compared to men. And so this points to the importance of these sex differences, because as we know, with the lower body water that women have per kilogram body weight, for the same amount of alcohol consumed, they will result in higher levels. And those higher levels may be contributing to those toxicities. So this slide now summarizes the possible mechanisms and outcomes related to these sex differences. As you can see, the several mechanisms have been proposed. The first, of course, being alcohol metabolism, where women have lower body weight and lower body water as a proportion to their body weight. And together that can result in a lower volume of distribution and so higher concentrations following the consumption of standard drinks. First-pass metabolism also shows differences between men and women. Women have lower gastric ADH and possibly slower gastric emptying. On the other hand, they do have larger liver volumes per kilogram body mass, but together they may be contributing to an increased bioavailability of alcohol, again, greater exposure. Sex hormones and other hormones have also been implicated. These may also have an effect on metabolism, but importantly, they might affect liver function and injury to various organs, such as with estrogen and its role in liver function. Growth hormone as well can be associated with increased hepatic ADH activity, and females, at least in animal models, seem to show altered secretion of growth hormone, and that can again result in greater acetaldehyde and other consequences. And then inflammatory processes such as the accumulation of endotoxin and activation of the Kupffer cells, these can also be modulators, and again, accumulation of pro-inflammatory cytokines and other mediators might put women at higher risk for liver disease. And while this slide is focused on liver disease, many of these mechanisms may be extremely relevant also for other pathologies associated with alcohol that seem to show a higher prevalence or higher risk in women compared to men. So finally, what I hope we've done today is reviewed with you the tremendous variability that exists in alcohol pharmacokinetics, particularly in the processes of absorption, distribution, and metabolism. And as you can see here in this spaghetti plot of breath alcohol concentration versus time, this was a study in a sample of 44 individuals who all received a standard dose of alcohol, and despite controlling as many of the experimental conditions as possible, we still see this 3 to 4-fold variability in the time course of alcohol concentration or exposure in the blood and therefore the brain. And so the consequences of this on its pharmacological effects or pharmacodynamic effects can be even more variable, and makes it really relevant and important to be able to understand and parse this variability. And what I hope we've also done is reviewed with you some of these factors that underlie this variability in alcohol absorption, distribution, metabolism, and elimination, both across and within individuals. And with that, we would like to thank you for your attention.